In addition to environmental factors like diet and antibiotic use, our genetics may also impact the gut microbiota. However, studies examining the effects of our genes on our microbiota have had mixed results, and many of the observed patterns are subtle. Twin studies are often used to understand the importance of genetics versus the environment in determining patterns we see in factors like disease. Take heart disease, for example. Remember, monozygotic twins come from the same egg and therefore have the same DNA, while dizygotic twins do not. The idea is that if monozygotic twins both have heart disease more often than dizygotic twins both have heart disease, genetics is likely playing a role. In contrast, if both monozygotic and dizygotic twins have heart disease equally often, lifestyle factors like diet and exercise are more likely to be the main influencing factors. A similar approach has been used for studying the microbiome as well. The idea is that if monozygotic twins have microbiomes that are more similar to each other than dizygotic twins, genetics must be playing a role. Sounds simple, right? Unfortunately, the data from these studies has not been simple at all. Some studies have shown that monozygotic twins are more similar to each other, while others have not. The data are conflicting. Another approach to determining how genetics might be influencing the gut microbiome is to put individuals with different genetics into similar environments and see if their microbes become similar to each other. If they do, environment is more important. If they don't, genetics are more important. These types of experiments would be difficult and potentially unethical with humans, but there have been a few experiments of this type with mice. In one, mice were placed with a different mother after birth. This mother had different genes than the mouse pup, but even so, the pup ended up looking more like the surrogate mother than its biological mother in terms of the gut microbial community composition. This suggests that environment is more important than genetics in determining the gut microbiome. Nevertheless, studies that look at the particular genes present in individual mice and people have shown that the presence or absence of a single gene can change the relative abundance of specific kinds of bacteria in the gut. This is particularly true for genera and species of bacteria. Gene presence or absence doesn't appear to have as much influence on the relative abundances of bacteria at higher taxonomic levels, such as the phylum. For example, lactobacillus relative abundances have been shown to vary with host genotype in studies of mice. Also, many of the gene-microbe interactions that have been detected involve immune function. For instance, humans with mutations in the MEFV gene get Mediterranean fever, an autoimmune disease. And these individuals also show changes in the gut microbiome. Similarly, mice without TLR5 gene function, which impacts immune function, have distinct gut microbiota. Many of the bacteria that have been found to be influenced by host genetics are also known to affect immune system function. So it seems that while the impact of host genetics on the gut microbiota may be more limited than the impact of other factors, it does exist.